So, hello, good evening. Today we'll be discussing a few problems as well as revising the concepts that were discussed in week 3 content. Okay, so it's 5 p.m. So I'll start the discussion. जैसे जैसे लोग join करते जाएंगे, people can subsequently join in. So, hello, Shri. Thanks for joining. So, I would like to ask you, in the previous week's videos, like in week 2, week 1, we were, told like how many types of mutations are there. So, can you name few types of mutations that you can remember right now? Shri, are you there in the meeting?
Yes, ma'am.
Yeah. So I was asking, can you recall? Yeah.
Frameshift mutation, somatic
Okay.
mutation,
Okay.
germline mutation.
Mm-hmm. Okay. Fine. Thank you. So you talked about frameshift mutation, frameshift, then somatic and germline. Then there were few types of point mutations as well, right? Which results in either missense mutation, nonsense mutation, right? Silent mutations. And we can also categorize these mutations as loss of function and gain of function. Correct? Then also there can be deletions, insertions, right? Insertions, deletions of few bases. That also results in a type of mutation. So these are all different types of mutations that were discussed previously, right, in previous week's video. But a new type of mutation that was discussed in week 3 is dynamic mutation, or also it is called repeat expansion. Correct? So yes, Yashika is joining. Okay, so I was saying, you know what is frameshift mutation, right? The ORF is shifted, means how the translation mechanism, the ribosome reach the sequence in the mRNA, that becomes changed, right? So subsequent addition of the amino acids, that is also changed. Then in somatic mutation, this mutation only happens in somatic cells, whereas in germline, it only happens in the sex cells, right? Like ovum and a sperm. So somatic mutations are not inherited. Germline mutations, they are inherited. And there are point mutations where there is permanent change in a single base pair, right? So it can either result in missense, like in the protein sequence, there's a change in the amino acid because one base pair change, right? Or instead of an amino acid, a stop codon is added, right? Because of which the protein sequence is now truncated. It has become shorter than the normal sequence. Or the point mutation is such that it is causing the addition of like it is substituting a base pair such that the amino acid remains the same, right? You know, codons are degenerate in nature, so because of that we see silent mutation. Then loss of function and gain of function mutation, right? So any of the above three mutations can be categorized either loss of function or gain of function. Okay, this insertion and deletions also refers to addition of few base pairs or deletion of few base pairs. So this, this, this. So these four types of mutations, they can be either categorized as loss of function or gain of function. So one of you, can you tell me like what do you mean by loss of function mutation and what do you mean by gain of function mutation? Just one line answer would suffice. Any one of you. Yashika, would you like to try? Yashika, are you there in the meeting? Okay, I think she's not there. So, so loss of function mutation means whatever change in the base pair sequence has happened, it has resulted in loss of the normal functioning of a gene or its corresponding protein. So the normal function has been lost. Okay, whereas gain of function mutation, whatever sequence change has happened, it has resulted in a gain of a novel function. Okay, so if suppose a protein is supposed to act as an enzyme for a particular substrate like substrate A, now because of gain of function mutation, it is now binding to another substrate, substrate B, right? So this novel function, this protein has gained, right? So it is gain of function mutation. So both of these loss of function and gain of function mutation, they have been identified to play important role for progression of any particular type of disease. Okay, so I hope loss of function mutation and what is gain of function mutation is clear to you all. Okay, because in week 3 lectures, sir actually discusses more deeply about this only in the first 2 lectures at least. And it is often seen that the loss of function mutation is recessive in nature. Okay, so the mutant allele can be masked by the presence of a normal or wild type allele. Okay, but gain of function mutation is often dominant. But it is not a universal thing. Reverse can also happen. Okay, so it is a general thing that people have studied while looking into the various kind of diseases. And another type of mutation that is discussed in week 3 is dynamic mutation. So in dynamic mutation, what happens? A repeat sequence, it the number of repeats, it multiplies. Okay, it multiplies as it is passed on from one generation to the next generation. Okay, so it is also called repeat expansion. Okay, so dynamic mutation is also involved in the anticipation kind of inheritance, which we saw like in last week's discussion in week 2. What is anticipation, right? So in those kind of disorders, dynamic mutation is often seen, okay, to playing an important role. Okay, now let's try to do one problem. Okay, so this is the problem that is visible to you all, I hope. Okay, and I want you guys to read it and tell me what will be the correct answer according to you. So the question states, given below are two DNA sequences and their corresponding transcribed amino acid sequences. So this is the DNA sequence and this is the corresponding amino acid sequence. Now one is wild type allele, another is the mutant allele. The mutant allele shows one base pair change. This is a point mutation, okay, which is highlighted in red. So here mutant allele A is the like substituted one, okay, and it converts the amino acid cysteine in the wild type. So this wild type protein cysteine is there, and in the mutant it is converted to serine. So you have to tell me what is the type of the mutation here. This is you're familiar with from your week 2 week 1 lectures. Can any one of you answer what is the type of the mutation?
Missense mutation.
Correct, you are correct. Thank you, Shri. The answer is missense mutation, correct? Because there is a single change cysteine has been changed with serine, okay, because of the point mutation. T has been replaced with A. The next question, steroid hormone, okay, steroid hormone receptor. This receptor is called X. Upon binding to steroid, it activates a set of genes that are involved in physiological pathway promoting muscle growth. Now, due to genetic mutation, the receptor X was able to interact with a few novel targets and activate their signaling cascade. Now, if some mutation has happened in this receptor X, normally it is involved in muscle growth. Now, due to mutation, it is able to interact with novel targets. Okay, so identify the type of mutation. So these are the options: dominant mutation, dominant negative mutation, gain of function mutation, missense mutation. So what do you think will be the answer to this question? Like the receptor X, because of the mutation, is able to bind to novel new targets. Normally, it is not supposed to bind to these targets, but due to this mutation, it has gained this function. So what is the answer to this question? Supratik, Yashika, what about you guys? Are you able to get this question?
Gain of function mutation.
Correct, you are correct. Thank you. So the answer is gain of function mutation. Okay. So here we are able to say gain of function mutation from the function of the mutated protein. Okay. And how, what type of mutation might have happened? Like it might have missense or nonsense, or a single point mutation or insertion or deletion. So because of these kind of mutations, like a corresponding protein function, from that we are able to say it is a gain of function mutation. Now this is another question. De novo mutation can be either somatic or germline. Okay, you're familiar with this, right? Now, which one of the following statements correctly differentiates between somatic and germline mutation? So these are the four options. A somatic mutation is heritable, whereas germline mutation cannot be inherited. A somatic mutation cannot be inherited, whereas germline mutation can be inherited. A somatic mutation occurs in the DNA sequence, whereas germline mutation takes place in the mRNA sequence. Somatic mutation is deleterious, whereas germline mutation is non-deleterious. So if you're aware, right, I have just explained what are somatic and germline mutations. So according to you, what is the correct answer then?
Option B, ma'am.
Correct, you're correct. Option B, somatic mutations cannot be inherited, right? Only the germline mutation can be inherited. Here's another question. Which one of the following statements are incorrect regarding loss of function mutations? Okay, so you are familiar with this. What is loss of function mutation? First option, these mutations may result in complete loss of genes function. Then these mutations result in either a reduced or complete loss of the gene's function. These mutations lead to novel or abnormal function of the gene, and these mutations are usually recessive. So you have to identify which of these statements are incorrect when you're describing what are loss of function mutations. So what do you think will be the correct answer? If you have difficulty in understanding any of the options, do let me know. So the answer here will be C, right? A loss of function mutation doesn't generally lead to gain of a novel or abnormal function of a gene. Am I correct? What do you guys think?
Yes.
Yes. So um, gain of function mutation is actually leads to a novel function, not the loss of function mutation. So the answer here will be C. And as I already told, loss of function mutation are usually recessive in nature. Okay, when recessive alleles are in homozygous conditions, only then they will be expressed, and they can either lead to complete loss of the gene's function or protein's function, or they can reduce its efficiency. Okay, and yeah. Okay. Now next question. So here, in this question, reciprocal exchange of chromosomal material between chromosome 9 and chromosome 22 lead to the formation of Philadelphia chromosome, correct? So you're aware, right? What is Philadelphia chromosome? And based on an understanding, choose the option that is true with regard to this anomaly. So you have to recall what a Philadelphia chromosome. Okay, so they are formed because of chromosomal material exchange between 9 and chromosome 22. So if you have to identify which is correct when you are typing normally. Is it caused by DNA double strand break, or does it lead to a gain of function, or is it chromosomal translocation happening, or all of these? What do you guys think?
All of these, ma'am.
You're correct. You're correct. So all of these contribute to the formation of Philadelphia chromosome. Okay, if you recall, and in the Philadelphia chromosome, the genes ABL and BCR, they fuse, right, and leads to the formation of a novel protein, correct? And this novel protein is often like involved in a leukemia, which is blood cancer. Okay, so this novel protein formation is like uh, here termed as gain of function, right? We are getting a new protein, and one segment of chromosome 9 is going and fusing with chromosome 22. This is chromosomal translocation, and it is only happening when there is double strand break. So all of these matches the events that are happening that leads to formation of Philadelphia chromosome. Now in this question, they're asking which of the following statements are correct when you are explaining dynamic mutations. Okay, so first, dynamic mutations depends on the number of repeats that are present in the gene. Then dynamic mutation may lead to anticipation. The anticipation is a phenomena in which disease is manifested at an earlier age in the subsequent generations, or all of these. So, if you remember the Huntington disease example, right, where dynamic mutation was involved, uh, how the Huntington disease was like inherited from one generation to the another. Can anyone means answer? Is the disease symptoms getting severe? Is it um, expressing early on the age? What was happening in the families where Huntington disease was there? So Pratik, you haven't spoken for a long time. Can you answer this question?
Yeah, I can.
Okay, go ahead.
The severity of the disease is inversely correlated with the age of onset of disease. So
Okay,
if some patient, sorry, some if some individual is showing disease at 60 years of age, in their successive generation, that disease will appear at least 10-15 years early. So in that way, it
correct,
processes. So with each successive generation, the age of onset becomes more earlier. So
correct. Hmm.
in that way, it is anticipated that severity will be increased and the age will be age of onset will be decreased in that case.
Correct. So that is what is observed in Huntington disease. So people saw that the cause of that particular disease was due to dynamic mutation. So I think now it is clear, right? What will be the correct answer for this question? So in dynamic mutation, what happens? There's an increase in the number of repeats. So in case of Huntington disease, CAG, this repeat, when it was greater than 35, right? The symptoms or the phenotype for that disease was expressing, right? In the normal case, it was less than 35 repeats. So with every generation, this like number of repeats was getting increased. So this statement is correct when we are trying to explain what is dynamic mutation. And in Huntington disease, dynamic mutation was involved, so it led to anticipation, which Supratik nicely explained what is anticipation. And here, this option is also correct. For this particular question, then option D is the is this clear to both of you, Yashika and Shri? I think it will be clear. If you have any doubt, yeah, great. So if you have any doubt till now in the previous questions, do let me know. Now, what are mutational hotspots? Okay, so mutational hotspots are those regions of the like DNA segment of the chromosomes where there is high chance of mutations to occur, right? For a particular type of mutation to occur. So some kind of mutations like repeatedly occur in those particular sequence of in the DNA, like in the chromosomes. Okay, because the environment surrounding that DNA segment is such that it is allowing this recurrence event to happen again and again. It is facilitating that. So when people have looked into multiple families which are not related to one another and saw like a particular type of mutation, whether this deletion or substitution or nonsense, whatever type of mutation, they saw that these mutations only occur at this particular segment of the chromosome. Okay, so then this term that this segment, if it is present in a particular family or a particular individual in that family, then the mutation will there and it will lead to a particular phenotype or a particular disease. Okay, so mutational hotspots are various types that people have identified. Okay, so it can be due to non-allelic recombination. Then there is founder's effect, which founder's effect, and a few bases within the sequence, they are modified in such a way that they attract recurrent mutations. So due to the presence of modified bases. So what do you mean by allelic recombination? Can any one of you say what do you mean by allelic recombination? Or simple, what is recombination? Any one of you? So this is a discussion session. It would really help, like if you guys, come like voluntarily come up and answer. So it will only benefit you. It will help you recall the concepts. So what do you mean by recombination? When it happens in a cell, what happens during recombination?
Uh,
Yeah, Subratit, thanks. Go ahead.
the equal distribution of genetic material from both the parents to give rise to a offspring. basically the offspring will receive equal amount of genetic material from father and mother. If it is done in an equal way, then it is called allelic. If it is not done in an allelic, like mismatch due to duplication, insertion, so it becomes abrupted the process, and then the process will be called non-allelic, and it can give rise to a range of uh, diseases.
Okay, so actually, Supratik gave the answer for what is non-allelic and allelic recombination. So I hope it is clear. Okay, but I was asking, like, what do you mean by recombination? Like simple recombination mechanism, and when does it happen in the cell? So whatever you have answered, Supratik, you are correct, and you have explained what is the consequence of this particular recombination. Okay, but I want to know, what do you mean by simple recombination? When does it happen in a cell?
In the meiosis, right?
Yes, recombination happens during meiosis. So in meiosis, there are two phases, if you recall, meiosis 1 and meiosis 2. So in meiosis 1, the tetrad is formed, right? How? The homologous chromosomes, the two homologous chromosomes, they pair like this. So recalling the basic concept that you have studied in class 11, 12 is also important. Uh, it will help you understand advanced concepts like this, what we are studying in this course. So I'm just recalling what you have studied already. So suppose these are two homologous chromosomes, one from father and one from mother. So usually what happens, if the one gene is present in this particular chromosome, then during recombination, and this is it, and in another chromosome, its allele is present. So this is the wild type, say, and this is the mutant one. Okay. So during recombination, what happens? Synapses is formed. Okay, where like this chromatid will, exchange some material with this one. This chromatid will exchange some genetic material with this one, like this. And during this, what happens? Say this wild type gene gets transferred to this homologous chromosome right here. So at the same locus, this is called the locus, right? Locus of a gene where both the alleles are present, okay, in the chromosome. So it gets transferred here, exchanged here, the better term will be exchanged, and here this particular gene get exchanged to this. So this is a simple recombination. And how proper recombination to happen, the alignment is very important, right? So this formation of this synapses and is very important. Then only all the genes and their alleles, they are aligned at the particular locuses. Now if it doesn't happen, okay, if there is misalignment, okay, and when misalignment can happen, when if this is the gene and it is flanked by repeats. So this is repeat R1, this is repeat R2, and in the clear this, and this is homologous chromosome. It also contains the same like allele of this particular gene, so it can be a homologous person or a heterologous person, and it also is flanked by the same repeats. So if it is properly aligned, then during recombination, some DNA material will be exchanged from here to here, like crisscross manner. Okay, and here also crisscross manner, here also crisscross manner. Okay, but if there is misalignment, then what can happen? Say this is R1. Okay, and due to misalignment, here's R1. R2. This is for the another pair of chromosomes for this. So here now recombination is happening between R2 and R1. Now they are similar sequences. They are not exactly identical. They are similar. Okay, but the alignment has happened in such a way that recombination can only happen in this manner. The alignment is governing how the recombination is going to happen ultimately. So because of this faulty recombination pattern, you know what will happen. So this is the gene copy. This is the gene copy. So at the end, one copy of the chromosome will have two copies of the same gene, and another copy, the gene is completely deleted. Okay, so this is the consequence of a non-allelic recombination. This is the non-allelic recombination that is happening. This is allelic recombination. Okay, so this is a very basic concept uh, that you are already familiar with from your class 11 12th knowledge of how meiosis and all is happening. A fault in that particular procedure leads to allelic recombination, non-allelic recombination, and because of that you get duplication. Okay, one allele is duplicated, means multiple copy of the same allele is present. Allelic duplication happens, or allelic deletions happens. So another copy of the homologous chromosome doesn't have that gene at all. Okay, and these type of sequences which are flanked by repeats, they are more prone to such kind of non-allelic recombination. That's why they are called mutational hotspots. As people have seen, when such kind of structure is present around the gene, okay, that gene is more likely to undergo either like that allele is more likely to undergo under either duplication or deletion, so mutation will happen. Okay, so this was clear, I think. If you have any doubt, please let me know. Now another thing that happens, the genetic drift. Are you aware what is genetic drift? This is also a thing that you have studied in your class 12 in evolution, how evolution happens. What do you mean by genetic drift? Can any one of you answer? Shri, can you answer? What do you mean by genetic drift? Drift, drift, and then genetic. The term itself explains what it could be.
Might be change in frequency of an existing gene in a population due to some environmental factors or a random chance that occurs.
Okay, so your answer was not very much clear because of some background noise. If did any one of you understood, can you repeat that answer for me? If any, I couldn't actually understand what she said because of background noise. Shri, would you mind if you can, write what do you mean by genetic drift in the chat box?
Chat option is not available.
Huh? What? You cannot write in chat box?
No, ma'am, can't able to access it.
Okay, what about, okay, fine. Can you then repeat your answer once again? So I'll try to understand it better this time.
Ma'am, change in frequency of an existing gene variant in some particular population.
Okay, so that is not exactly genetic drift. Okay, so what happens is there's a big population, okay, and they choose a few members of those people, they choose to migrate to another location, like due to some dispute or war or something like that, they choose to migrate. Now the genetic material that these few people are going to contain, so they because of that, and they are means populating another area, okay, that is geographically completely different. So there is a flow of genes, okay, from this population A to another isolate area. Now, genetic drift doesn't say that this area, the new area, it has to be completely new. Okay, it can have other people as well. So what has just happened is new pool of genes that from population A has migrated to another population, say B, because few people decided that they want to migrate. So there is a flow of genes. Am I correct? Or did you guys understood what I tried to say?
Can you repeat once?
Okay. So if you Google it just simply, okay. So here the definition is exactly what I think this she said, right? That the change in frequency, okay, and due to the change in frequency of an existing gene variant in a population due to random chance. But what does it actually mean? If you are given population A and population B, what actually has happened? So in genetic drift, there's a population A, okay, and the few people they have decided that they want to move to another location, okay, and in that location there is some another population that is living there, okay, and these are people of the same species, the species are same, they are carrying same types of chromosomes and all, and because of this migration to another population, uh, and they have migrated to population B, and these people are marrying with people with the who are already living in population B. So because of that, that particular the whatever genes, the alleles, mutant alleles they were carrying, their frequency, or you can say their number actually changed. It can multiply, right, within the population B when they were marrying and mating and giving rise to new people. So because of this, selection, natural selection, and all happens. Do you remember this process, natural selection, when nature actually selects when a new particular. Let me charge my laptop. Okay, fine. It is now charging. So as I was saying, that nature actually selects the presence of a new phenotype that has arisen because a new set of people carrying some mutant alleles, and when they have married few people in the new population, these alleles are multiplying, right? The frequency has increased, the number of these alleles have increased within this population. So new phenotypes are arising within the population B. The nature can select which is favorable for surviving in that particular environment. So if I just read from one article in the internet, it says that genetic drift contributes to the natural evolution of the species, and it leads to the fixation of new alleles that have been recessively fixed in the previous individuals, and the development of new features in the population. So I was just saying that, right? A few people from population A carrying some alleles, they migrated to another population B. When they married and all, these alleles, they multiplied, their frequency increased, and they led to the development of new feature within the population B. And the nature selected, right? Which phenotype is desirable, one phenotype is not desirable for surviving in the environment. And after thousands of years, you get a species who are having this new phenotype, speciation, or it can also lead to new species generation. Actually, not only change in the phenotype, but it can also lead to new species. Am I clear?
Yes, ma'am.
Okay. So that definition that you told is correct, but it doesn't give the complete picture. Okay, what is actually happening in genetic drift? Now, genetic drift is, you can now understand, it is completely different from founder's effect. Here, few people in population A, they're migrating to a new area, okay, where there were new humans before, and now these people are the only humans that were there in that particular land, and now they will marry and have children. So because of which few mutant alleles they carried with them, they can multiply, or de novo mutation can also happen. So that's how genetic drift and founders effect are completely different. Although sir didn't discuss about genetic drift, but I thought when studying some other literature, and you may stumble upon these two terms where they are talking about evolution. So genetic drift, like, it leads to selection of the desirable characters, but founders effect, you can say, can lead to the expression of the diseased phenotype as well. Okay. And when founders effect happen, since the mutant alleles, if they were recessive in nature, so in heterozygous people they didn't express. Now due to mating and all, they are expressed in homozygous manner, so they lead to expression of a particular disease, right? And in that isolate population, if people carrying that particular disease, if they mate more and more, then the chances of for that particular disease to arise become higher. So and when they check the DNA, means the sequence of the mutant allele, they saw how it is getting passed on. People were homozygous for that mutant allele. Ultimately, their children will be also homozygous, right? So in that particular population, that disease is more recurrent. So that place can be called a hotspot for that particular mutation. Is it clear? Is it clear? The difference between genetic drift and what do you know founders effect? Why founders effect actually leads to creation of mutational hotspots? Is this point clear? Supratik, it was not clear to you previously. Now it is clear to you, I hope.
Yeah, both are interrelated, but could be a slight difference in between these two.
Correct, correct. They are slightly different, but it both happens due to migration of people.
Understood. Genetic drift is a normal phenomenon which could happen easily at any time, but founder effect is a particular phenomenon which can leads to new I think new population, can bring new mutations.
Correct. So if you look at today's scenario, there is no land where man is not there. So founders effect is likely possible again, right? But genetic drift can happen, right? Indians go abroad and marry a foreigner and have children and all. So it is happening all the time, right? A South Indian marries a Bengali person, something like that. Genetic drift is happening. But founders effect, it has happened previously, like few hundreds of years ago. And now, whatever their children and all the diseases that they are having, the people are identifying that they happened because of founders effect. Okay, it led to um, presence of both the recessive alleles in their children, because of, and those recessive alleles, they're carrying the mutation which is either a loss of function mutation or a gain of function mutation, and leading to a disease. Then another reason, like why few DNA segments they are mutational hotspots, or why mutations are more and more recurrent, is due to presence of some modification. Now, modification can be many types, but like as sir explains an example, one is methylation, means adding of a methyl group. Okay, where it is added at the base. So it is seen that these methyl groups, they have more affinity to get added on the cysteine, right? Cysteine base of the DNA. And because of that, because addition of that methyl group, uh, the base, if it is acted upon by some enzyme, deamination enzyme, it can convert to T, that is thymine. So because of the presence of this modification, that is methyl group on cysteine, we are getting a point mutation kind of a thing, right? One base is changed another base to another base, and this point mutation is recurrent, is repeatedly seen because the presence of this modification. If this modification is not present in that DNA sequence, this thymine conversion would not have been recurrent, and it has been linked with various kind of diseases as well, right? Like Rett syndrome. I just recalled Rett syndrome. People have seen that. Okay. So I hope till now it is clear to you. Like why some kind of mutations are hotspots. Okay, means those mutations occur repeatedly in different individuals who are carrying. So what kind of mutation that person should carry, depending on that, it is recurrent, and it is recurrent in the population. So based on this concept, here's a question for you guys. You have to answer which of these options exactly explains what do you mean by mutation hotspot. So these are prone for mutational events. That then option B says it promotes mutation in a DNA that comes in its vicinity. Option C is it has a rich source of mutation, or all of the above. What do you guys think is the correct answer for this question?
All of the above.
Okay. So why do you think it is all of the above?
Option A, prone for mutational events.
Okay, you're correct. Thank you, Ashi. So hotspots, mutation hotspots, just now I said that these areas are very much prone for recurrent mutations for happen. And what kind of recurrent mutations, that I've already explained, right? So option A is the correct answer. It is not like they are rich in mutagen. So what are mutagen? Mutagen are basically sources which cause the mutation. Like you have heard UV, right? UV can cause DNA damage. Then there are few carcinogenic chemicals like ETBR, right? Which is used in electrophoresis. If you guys are from biotech background, you would know. So ETBR also causes, DNA mutations. So these are mutagens, the sources which that actually lead to a mutation. So it is not like these are rich in mutagens, and uh, the mutation only it happens at the coding sequence. Okay, and because of which we are seeing direct effect, right? As a loss of function or gain of function of particular gene or a particular protein. So the mutation is not like it happens near some sequence, like in non-coding sequence, something like that. It is not like that. Okay, so the option A is the correct answer for this. Is it clear, this question, why option A is correct and none of these other options?
Yeah.
Okay, thank you. Now this is another question. The process marked as A in the image given below results in generation of recombined chromosomes. This is your parental chromosomes, A, maternal and paternal, and you have to identify what is this process A. like from the options given. What process leads to formation of a recombined chromosomes like this? Can any one of you think and give a possible answer for this question?
Allelic recombination.
You're correct. So here proper alignment is there, right, of the both the alleles. So because of which allelic recombination is happening. Misalignment is not there, so non-allelic recombination is not the option. Okay, so non-allelic recombination leads to duplication of a gene or a deletion. Okay, and here any NHEJ is basically a DNA repair mechanism, so it is not at all related to this topic. Then another question: non-allelic recombination does not lead to what? Duplication of a chromosomal segment, deletion of a chromosomal segment, recombination of chromosomes, then non-disjunction of chromosomes. What do you guys think is the correct answer for this question?
C. It's the C.
Option C. Okay. Why do you think option C is correct? Recombination is happening, right?
A recombination in the sense normal, I think, because both the things, if I can check out all the options, duplication can happen in case of non-allelic recombination, deletion can also take place. Non-disjunction also is a major part of various, I think, translocation events. So I think it should be the recombination of chromosomes.
What about others? Do you guys agree with him? Arshi, what about you? I think you unmuted yourself. You are going to answer, I think. Do you agree with Supratik?
Ma'am, I'm confused between duplication and deletion.
Okay, acha. So here you're being asked what does not lead to what does not happen in non-allelic recombination. So duplication does happen, right? If there is misalignment of the homologous chromosomes, then a particular segment of DNA, a gene say, it can get duplicated during non-allelic recombination, and in another chromosome it can get deleted. So if the alignment is not proper between the homologous and non-homologous chromosome, then both the events can happen. Are you getting what I'm trying to say? So here I actually drew. Okay, so let me say in this figure. Okay, in this figure that is given here. So if the alignment is not like that, if suppose this is the I'm drawing the maternal section, this is the say orange, this is the gene, this is the orange repeat, and this is the father one, paternal one, and this is the green one, this is the gene. Let me type green with GR. This is GR. Now the alignment is such that when recombination happens, okay, between this repeat and this repeat, okay, then in one homologous chromosome, this is orange, resulting, this is the resulting what I'm drawing. This is gene, this is another gene, this is another orange, right? So in one homologous chromosome, these two gene segments get combined, right? And in another, let me draw here, what will happen? Here green is there, right? Sorry, here it will be green and it will combine with, sorry, it will be orange and here it will be green. So it will combine like this, right? O, G, here the green is there, right? And this is green and this is another green. A segment like this will form. Whereas in another segment, only the repeats will be present. I may not have drawn it correctly right now, okay, because recombination thing can be confusing for me, but that is what I'm trying to explain. Due to the misalignment of the homologous chromosome, one pair of the homologous chromosome can get both the copies of the gene, whereas in another, only the repeats are left. The single copy of the gene is now missing, deleted. So that is what happens in non-allelic recombination. So these two options are correct for non-allelic recombination. But here you are asked what does not lead to non-allelic, what does, it is not the sequence of the consequence of the non-allelic recombination. So the confusion is between C and D. Okay. So Supratik answered that correct answer will be C. So let me answer. The correct answer for this question is D. Okay. What do you mean by non-disjunction? If you recall, this is the mother cell with a pair of homologous chromosome at the beginning of meiosis 1. In meiosis 2, what can happen? Normal case. At the end of meiosis 1, these two homologous chromosomes get separated, right? The synapse gets broken. The recombination has happened. This is the end of meiosis 1, this stage. Then it enters to meiosis 2, these two cells. And at the end, you get 4 cells where I have not drawn it nicely. Let me draw it better. Where only one sister chromatid is present. This is the normal case. But what happens at abnormal cases? At the end of meiosis 1, you can get 2 daughter cells, one carrying either both homologous chromosome and another does not have homologous chromosome at all. This is the abnormal case. Or what can happen is, say at the end of meiosis 1, the homologous chromosomes, they have distributed equally. But at the end of meiosis 2, the sister chromatids, they did not distribute equally. So here there is no sister chromatid, and here is sister chromatid, this is sister chromatid. Now when this gamete goes ahead and fertilizes and another gamete, male or female gamete, then that person will be lacking one whole chromosome. So that is non-disjunction chromosome, non-disjunction. That is the event of non-disjunction, where there is another case called translocation. Okay, and I think Supratik have confused between these two. In translocation, if this is a chromosome, okay, a single chromosome, and this is another chromosome, there's a double strand break here, okay, and say here also double strand break, because of which the DNA content is swapped. This is a translocation. This is the translocation that happens between chromosomes. Got it? So for this, so non-allelic recombination for this question, it does not lead to non-disjunction, but it does lead to recombination process. Is it clear to you all? Or is there still any doubt?
Yeah, what will happen to that? Like you explained, like it's a splitting up a fragment and joining up a fragment to other chromosome in case of chromosomal translocation. But in non-disjunction, how it is different? How it can be differentiated from the translocation.
Okay, so non-disjunction, as I told you, so at the end of meiosis 2, okay, normal cases, each cell will have one sister chromatid because of which they are all haploid, right? You agree? Now this is a normal case. Now if non-disjunction is happening, then it might be possible that this sister chromatid during metaphase, it didn't separate. Okay, it didn't separate towards the two poles that is happening in anaphase, because of which one daughter cell is formed where it doesn't contain the chromosome, it lacks one whole chromosome. The consequence you can see in this Klinefelter syndrome, if you remember, right? It happens for the sex chromosome. Then there is also Turner syndrome, then Down syndrome, right? There's a gain of whole one chromosome. It was happening because this particular gamete, say, fused with another gamete that is coming from parent, right? Then it is normal. Okay, then it is normal. Now suppose this particular that if it fused this gamete, if it fused with another gamete containing both chromosome, then the chromosome number will not change. But if suppose now this chromosome, if it fused with a normal gamete containing only one pair of chromosome, then there will be trisomy, right? So this is
So
the yeah.
So what I understood, this non-disjunction can leads to both loss of a chromosome or also gain of a chromosome.
Correct,
Both
correct,
cause like genetic disorders.
correct. Complete whole chromosome. But in translocation, segment of the chromosome is lost. Okay, either it gets deleted, or in Philadelphia chromosome, as you saw, there was exchange.
Yeah.
So both can happen.
Yeah.
Now moving on to the next question. So there is a small tribal population on an island that shows high frequency of a genetic disorder. Which of the following may not explain the high occurrence of this disorder? Will not explain, okay? May not explain. One is founder effect, then continuous practice, anticipation, and genetic drift. What do you guys think? So there's a small tribe population, okay, and they're living on an island, so they're isolated from the rest of the world, and they have a high frequency of a particular genetic disorder. Means there's a mutant allele that is being expressed more and more, because of which many individuals have that particular genetic disorder. Now, which of these following examples will not explain why there is a high occurrence of this disease? What do you guys think the answer to this question would be? Arshi, would you like to try?
Genetic drift.
Okay. So you are guessing genetic drift, thinking that this tribal population are the founders, right? Or okay, because they have moved to an island. So genetic drift, as I already explained, it may happen that in that population, whoever brought that particular mutant allele with them from earlier population, So they settled in an island where people already existed. Okay, so due to genetic drift as well, that mutant allele has come into that population in that island, and they're marrying and all. So the children are having this particular disorder because of homozygosity of that recessive mutant allele. Genetic drift can explain this particular um, disorder. The question asks, what does not explain? So if the other case would have been that island was completely human, there was no human at all, okay, in that particular island, then founder's effect could have happened, right? And if neither of that case, no drift, no migration of the people are there, even if people just married their siblings, just like what happens, you have studied, like you have seen videos as well. British royal families, in order to preserve their wealth, they used to marry their siblings or cousins and all, right? Even pharaohs used to do that. So contiguity practices also leads to the expression of the mutant allele. Okay, homozygous condition, then the disorder happens. But what does not explain is anticipation. The correct answer to this question is anticipation. Do you guys agree? Do you guys agree? Do you guys think that I'm correct? Why the
Yes, ma'am,
anticipation
it is
would
anticipation.
correct?
Because in anticipation, every generation the frequencies increase.
And it is due to dynamic mutation, correct? It is a dynamic mutation. It is happening, and it doesn't affect the frequency of the particular allele. It affects the frequency of the particular repeat in that particular allele, right? So anticipation is the answer, which is not explaining why more and more people are having this particular disorder. Anticipation stays within the family. Okay. Um is that correct? Do you guys why do you guys understand why the answer is anticipation? Others?
Yes, ma'am.
Great. Now moving on to the next question. So here in few 3-4 questions, I'll just revise about PCR, okay, and sequencing. So these things I think were there already in week two content, but these are bit challenging questions for you guys. So I think you guys can do it. Okay. So first question is, PCR can be used for which of the following processes? First, cloning a segment of a non-coding region of the chromosome, cloning a coding region present in the processed mRNA. It can detect mutation, all of the above. So PCR, you have to guess which of the following options PCR can be used. What do you guys think? Shri, would you like to answer this question?
All of the above.
You're correct. You're correct. So I think it is self-explanatory why the answer is all of the above, right? Or if it is not clear, I can explain to you guys. Please let me know if it is not clear to any one of you. Okay, if you're still shy, then I'll explain anyways. So PCR is used for amplifying any DNA or RNA sequence. DNA and RNA only differs in one base, right? So if you design a primer in such a way that it can bind to a particular sequence, okay, nucleotide based sequence, then PCR can be done. And all you need is a template sequence, which can be DNA or RNA. You need a polymerase enzyme, right? You need few primers and few nucleotides. And then you have to give temperature cycles, right? So was only using these things you can detect whether a particular sequence is having a mutation or not. You can do PCR for both the wild type or the mutant type and then compare, right? You can then PCR product can be run through gel electrophoresis and you can see where the band, where is the band you're getting. You can you can clone the mRNA sequence, and you can also clone the coding or the non-coding, doesn't matter, as long as it is a DNA sequence, base sequence, which is having ATGC or AUGC. Okay, so all of these options can be, for all of these options, PCR can be used. This is another question. I'm reading it aloud. So you plan to develop a microsatellite marker from a genomic locus bearing a polymorphic repeat of CGG triplets. Okay, the sequence surrounding that repeat region is made available to you, and same is shown in the schematic diagram given below. This is the sequence. Okay, the 5' 3' both are given, and so we have to identify which of the following is the correct forward primer and reverse primer respectively, which can be used to amplify the repeat region. So you have to identify among these four options which of these you can use for amplifying this sequence. So as I already told, for PCR you need both forward and reverse primers. So what do you guys think will be the correct option here? I'm like zooming it out for you so that you can carefully see the sequences, okay, the given sequences, and try to match the given options and guess any answer. If you're even if you're not understanding, just guess. It's not an exam, so it's fine. So do you recall where forward primer is ligated and where reverse primer is ligated?
Ma'am, option D.
Option D. Okay, why do you think it is option D? What made you guess? Yeah.
5' CAG is complementary to the 3' GTCATA strand.
Okay, okay. So if suppose I'm not writing the basis, this is your 5', this is your 3', this is your 3', this is your 5'. So where would the 5' means where would the forward primer will be ligated? In the first strand and the second strand. The first strand, second strand, where it will be ligated. If you give a forward primer, where it will be ligated? And is it going to ligate? Say this is your left side, this is your right side. Now you can answer. Where the forward primer will be ligated? Is it going to ligate or is it going to complementary bind on the left side or right side with the first strand or the second strand?
Left with the first strand.
Okay, so if it is going to go here left on the first strand, so correct answer is D only. Okay, Arshia has given the correct answer, but you can see that the forward primer is not complementary. Is it same? Right? It is same, exactly same, the sequence. So you should remember forward primer goes here. 5 prime to 3 prime, so that it can add more and more nucleotides and cause polymerization reaction in this direction, 5 prime to 3 prime end for the second strand. Whereas the reverse primer goes here, right side of the first strand. Okay, and here if you check, and if you if you reverse the given sequence of the reverse primer and right here, and if you complementarily bind on this side, you can see A T is complementary, right? T T A A. So this is exactly the same as the 5 prime end on the right side of the second sequence. So whenever you are doing PCR, you should remember to say you want to amplify this region, right? So you will choose the flanking sequence, which will be around 15 to 20 base pairs. Here they have shown very less, but for specific binding, we go for a bit longer sequence, but not too big, okay? 15 to 20 is optimum, okay? And the forward primer is the sequence of the forward primer is exactly the same as the 5 prime end of the first sequence, okay? Whereas the sequence for the reverse primer is the complementary of the 3 prime end sequence on the right side of the first. If you remember that, you can just simply look at your double-stranded DNA, okay, and choose 15 to 20 bases, 5 from the 5 prime end and as well as the 3 prime end. And even if you have just the 5 prime to 3 prime end sequence, you don't have this second one, you can design your forward and reverse primer. The forward primer sequence will be exactly same, okay, same as this sequence, and it will reverse primer, the sequence of the reverse primer, it will be complementary to this 3 prime end sequence. That's it. You have to do that's it, and you can then ask any company, or you can do it yourself to synthesize a new DNA strand, a small DNA strand, okay, and use it in your PCR. So people don't actually teach these things, how to design forward primer, reverse primer. But I'm glad that I found this question so that I could explain to you guys. Is it clear to everyone?
But the reverse primer, how do you design reverse primer? Because synthesis always takes place from 5 to 3. Is it the reason why it is always attached to the 5 end of both the strands?
Actually, your question is not clear. Can you reframe it and like tell the question again?
Suppose the synthesis of DNA and that transcript synthesis always takes place from 5 to 3 n. So is it the reason why the primers are added at the 5 prime end of whether it is the leading or the lagging strand?
Yes, correct. You're correct. So see, in this like whatever image that I have drawn, here you can see that this is the forward primer, right? It is complementary to the second strand of the DNA, and here the nucleotide bases are being added and ligated. That is polymerization reaction that is happening, and this is the reverse primer. And this reverse primer is also due to the presence of the reverse primer, the polymerization reaction can grow from 5 prime to 3 prime end in this direction. Okay, so reverse primer is helping the amplification of the first strand, whereas the forward primer is helping the amplification of the second strand. Forward primer, why we call? Because it is added the beginning, left side. This is how we read, right? This is how we read anything. So it is added at the beginning, whereas the reverse primer, it is added at the end. So don't get confused with the nomenclature, forward and reverse. But remember, if you are given a DNA sequence, okay, and now you're going to do PCR with it, how you're going to make the primers for it, the both forward and reverse primers. And you need forward and reverse primers because the DNA is double stranded, right? And you want DNA replication to happen for both of these strands. So you need these primers are adding what? These primers are giving 3' OH group to which nucleotides can be added. If you remember, phosphodiester bonds has to be formed, and it can only be formed if the OH group is present. So because of this, these bonds are made, and the sequence is, completed, right? The sequence will obviously complementary to the template, whatever template you have chosen. So is it clear?
Yeah,
The
understood. But you say that forward primer always complementary to the lagging strand or something you said. I missed that.
So here, when you're talking about PCR, we don't use the term first, the leading and strand and lagging strand. Okay, so these terms are only applicable when you're, talking about DNA replication, which is happening inside the cell. PCR is just mimicking uh, what is like happening in inside the cell. We are doing it artificially in a machine. Some thermocycler machine is there, okay, and we're adding all the ingredients and asking the process to happen. Enzyme is given, so it is a catalysis process, you can say. um, Simple polymerization reaction will happen in presence of an enzyme. So don't use the term leading or lagging strand here. Just you think that there are two strands of the DNA, right? One is from 5' to 3', another is from 3' to 5', and you are asked to design a forward primer and reverse primer. Okay, so as I told you in this figure, where what will be the sequence of the forward primer? It will be exactly the same as the 5' end sequence, okay, of say the first strand. Okay, this, it will be exactly the same, because of which this forward primer can go ahead and complementarily bind with the sequence which is running from 3' to 5' because of that. And the reverse primer, it will be complementary to this 3' end sequence if you read from the 3' end. So if you're giving just one sequence, you're not given only the sequence reading from 5' to 3' end. You're not given this sequence. If you're not given this sequence, you can design your primers by looking at this sequence only. That is the message that I'm trying to relay.
Yeah.
Okay, I think it's clear to you all. Okay.
And is there a
Now,
coding strand we call her? We
okay,
call anything as coding strand.
okay. So again, when we talk about transcription, okay, transcription. So there are two strands actually. People give name for these two strands in context with a particular process. So the coding strand, it usually contains the DNA sequence which codes for the particular gene. Okay, so if you match this sequence of the mRNA, resulting mRNA from a DNA sequence, you will see that its sequence is completely same as the 5' to 3' end. Okay, the coding strand is not involved, it is not acting as a template. Another term, template strand, people use. Okay, then other than template, people use sense strand. Okay, then there is nonsense strand. So that why these terms are used depends on the context, depends on the process. So when people are doing transcription, okay, the template strand is there, which is from 3' to 5', okay, and from which the complement it is being used as a reference for adding the nucleotide bases, okay, in the mRNA, resulting mRNA. That's why it is called template. Whereas if you check the DNA sequence of the coding strand, it is exactly the same as the sequence in the mRNA. All right. Now this segment of the mRNA that you have made, it is coding for particular protein, right? So since this sequence is same exactly as the 5' to 3' end, so that sequence is called coding strand. Got my point? It can get a bit confusing.
Yes, ma'am.
But you have to understand in what context that terms are being used.
Okay, ma'am. Okay.
I think my screen is not visible. Let me share it again. Okay, now it is visible. So this is another question that is based on sequencing, okay, DNA sequencing, and it is called Sanger sequencing, which we already talked about in the last week. So in the assignment problem, you were given a similar question like this. So it is a simple version of that assignment problem only. So here in this agarose gel, you're given means a Sanger sequence link. After they have done Sanger sequencing, they have run the agarose gel. Okay, and using this image, you have to correctly identify the sequence of the DNA. Okay, so I'm not going to explain Sanger sequencing again because I've already done it in the previous session last week. So it is just an example of that concept. Using this image, you have to guess what is the sequence of the DNA fragment that you have given for sequencing. So what do you think will be the correct answer? Let me, make it bigger so that you can read it. You have to read this gel, agarose gel. Okay, and I've already told, I think you must be familiar how to read it. The way you read it will give you directly, like in one line, you can get the sequence. That is how Sanger sequencing method works.
Ma'am, option D.
You're correct. Thank you, Arshi. You're correct. Exactly, you're correct. So did everyone understand why it is option D? So we have to always read from bottom to top. We have to read in this direction. Okay. Why? Because DNA is negatively charged, right? And when we give positive charge, it will try to migrate towards positive charge, positive end. So the DNA strands which are small, right, they will migrate fast towards the positive end. Whereas the DNA strands which are longer in length, they will still remain towards the negative end. So when you are reading this agarose gel, we should start reading the sequence of the small DNA fragments. Okay, so here this is the lowest one. I will read just like this: A, then it's a thoda upar is C, right? I've read this. Then G, correct? Then C, 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 right? I'm climbing up the ladder. That's all. Then there is G, right? Then there is A. Again there is A. We have to climb up the ladder and check each column. Then T, okay? Then again A. Then G, C, C, C. Then A. Then again G. Then A. T, 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 T. Okay? So if you see options D, A, 3C, G, A, T, A, 3CC, GAT. So I have arrived at the correct answer. That is option D. So I've just read this gel in a ladder manner, okay? And we have to check each column. Is it clear to you all how I've read this agar gel which we have obtained, okay? And then after reading this gel, I'm able to get the sequence of the DNA which was unknown to me previously. Is it okay? It is clear to everyone? I was not able to show you guys in the last week as I didn't have any particular example, but I thought it will be today I can show you guys. I hope it's clear, right?
Understood.
Thank you. Now, moving on to the animal models. Okay, so why do we need animal models? Animal models actually help in understanding any disease pathology, or pathology means what are the symptoms of that particular disease. Then also it helps in understanding what is the genetic basis of that particular disease. So people use different kind of animal models, right? Like zebrafish and all, you must have heard. Zebrafish has been genetically modified for, it can give lights, different kind of lights, luminescent, bioluminescent zebrafish and all. So for humans, for studying human genetic disorders, people use mice generally. Okay, and in all very good papers, people prove it. Whatever results they have gotten in vitro, means in lab setup, they try to mimic those results in vivo using mice models to give much more concrete proof, okay, for their work. So how these mice models they are created? Okay, so people generally use either a transgenic mice or a knockout. Okay, now knockout can be global or it can be conditional. You guys have seen the lectures, so you would know what I'm trying to say, right? So for transgenic mice, we are adding or inserting a gene of interest. So using transgenic mice, we can study gain of function mutations. Correct? Whereas we knockout, right? Knockout means deleting, right? Deletion. So we are deleting a particular segment of a DNA. It can be one exon or a portion of an exon or 4 or 5 exons of a particular gene. So we are deleting it, and it is generally helpful in studying loss of function mutations. So if you know, okay, that a particular genetic disorder is due to gain of function mutation, where a protein or a gene has gained a novel function, then you can actually create a animal model. Since you cannot experiment with humans, you can create a mice, okay, where you first cause that particular disease to happen by adding this particular mutation, okay, and then you can check all the treatments that are possible, like you discover some drug or something like that, or another group is working on a drug, so you can ask those people to give their drug, and then you can inject into that mice and see how the symptoms, whether symptoms have improved or whether it have become worse. Okay, so you are also checking for particular kind of treatment for that particular genetic disorder. Okay, so same goes for knockout as well. Now knockout can be global, means you can delete that particular gene in all the cells. Okay, somatic cells, then germline cells, then cells of all tissues, whatever tissues they are, like brain, heart, muscles, whatever. So in all cells, that particular gene has been deleted while creating a global knockout mouse. Whereas conditional, knockout, what happens? You are deleting a particular gene, or you're causing the loss of function of a particular gene in a particular type of tissue only. Okay. Now, if you are aware, or if you have seen that a particular gene, okay, is having a dual role, one that is causing embryonic development, and another it is causing some important function in in particular tissue. So if you do conditional, if you do global knockout, then due to the other function that helps in embryonic development, your the fetus will not at all mature at all. Okay, the embryo will not mature, so you will not get the knockout mouse at all. So in that case, the conditional knockout is a much more better option, because you're uh, deleting that gene in only that particular tissue. Okay, you're not deleting that gene in any of the tissues or any of the cell types other than that tissue specific cells. Okay, so you have understood why knockout mice is useful, why transgenic mice are useful. Okay, and conditional knockouts, they are like created, or say knockout, when you do knockout or loss of function type, when you try to incorporate a loss of function mutation, use the recombination mechanism. This is, I think you recall, right? uh, these things from the lecture. You use recombination mechanism. Here you actually um, add the gene by using some vehicle, like it can be a plasmid, or it can be a virus, retroviruses and all. So that particular vehicle carrying your gene of interest, okay, and that gene of interest can have a promoter upstream of it, okay, and you have created yourself a recombinant DNA, it is also called, right, where plasmid along with the gene of interest. So now you're inserting it into the nuclei, okay, of the um, fertilized zygote, where the nucleus of both sperm and egg are yet to fuse. They have not fused yet. So there you have this injected. Now you have no control where your gene of interest will get incorporated in the chromosome. It can get incorporated anywhere. Okay, so you have to do it for multiple samples, and then when you grow, then when you allow that both the nucleus from the sperm and the egg to fuse, right? The fusion process, there is exchange of DNA and all DNA segments between the mother and the father. One, right? Cell division will happen. It will create multiple cells. So ultimately, you will get some progenies, right? Some daughter mice and all. So there you have to check um, the phenotype, okay, the phenotype of that particular mice. If the gene of interest have expressed, you will get the desired phenotype that you were expecting at the beginning of the experiment. So that is how, in brief, that is how you create a transgenic mice. You deal with only one gene which is added. Now in knockout, you are going to delete that particular, either complete gene you're going to knock out, or you're going to knock out a small segment of that gene, and you're going to use a recombination mechanism. Okay, so you create again a DNA transcript. Okay, say in a plasmid like this. Okay, this is a plasmid, and this is the DNA segment of mouse, and this is the plasmid carrying, say, one DNA segment which is complementary to this DNA segment. Okay, you're designing this particular, you're designing this, okay, and you're adding another segment within the plasmid which is complementary to, say, this segment. So what happens in recombination, the similar sequences or the allelic sequences, they align, right? And you want to delete, say, this particular gene segment, and recombination will happen here. So when recombination will happen, and in your plasmid you have added some gene, right? Say neomycin gene. With this gene is going to give resistance towards the neomycin antibiotic. Okay, so here recombination is happening at these locations. Okay, so now you can guess, because of the recombination, this particular DNA segment will come here, right? And this particular DNA segment will go there. So what the cells, the embryonic, we are doing this inside mouse's embryonic stem cells. Now, embryonic stem cells are actually pluripotent stem cells. These stem cells can give rise to all the organs of the mice, and they are present in the blastocyst structure, if you recall, right? From the embryo, morula, then blastocyst, then the blastocyst, there's trophoblast, inner mast cells, and within the inner mast cells, you can find the embryonic stem cells. The scientists first have to isolate those embryonic stem cells, and within those embryonic stem cells, they have to, transduce this particular DNA segment. Okay, and when those cells, those embryonic stem cells where recombination has happened successfully, recombination will only happen during cell division, right? During cell division, it will happen, meiosis, it will happen. So when it has happened successfully, the cells which are carrying the neomycin gene will be selected, right? Positively. You know what is selection? So it is acting as a selective marker. So in presence of neomycin, these cells will grow properly. And those cells where recombination has not happened properly, those cells will die in presence of neomycin, right? Because it is an antibiotic. So uh, now you have selected these cells. Now in your construct, for for becoming double sure that where this region of the DNA segment of the mice where you want the recombination to happen, whether or not recombination has happened only there. Okay, so it can happen randomly. As I told you, in transgenic mice, the gene can get added anywhere. Similarly, the recombination can happen randomly if these two sequence, if they are similar to any of the sequences. Now, in order to avoid that kind of scenario, you can add a toxin gene. Now, this toxin gene uh, is not getting um, added. Okay? So this toxin gene is not here, right? Whether neomycin should be present, it is not here. It is away. It is away from this particular segment. It is away from there. So if recombination has happened somewhere else, then this toxin gene can get added into the mouse's embryonic stem cells. Okay. Now, if the recombination happened only at this segment of the DNA where you intend, this toxin gene will not get added. Okay. Only the neomycin will get added because the pattern, how the recombination is happening. So in those cells where recombination happened at the undesired location, those cells will carry the toxin genes, because of which the toxin will be expressed and the cells will die. And the cells where the recombination has only happened at this particular location where you desired, toxin gene will not get added in the mouse's embryonic stem cells. Okay, the mouse's DNA segment, and these cells will still survive. There will be no expression of toxin. So because of, so ultimately whatever cells you will get, you will find that the desired DNA segment, it has been deleted. Now if you are doing for all the if these embryonic stem cells are then getting added into the blastocyst and then forms embryo and all, so all the cells of the tissues will have this knockout. But in conditional knockout, what happens? Here also recombination mechanism is going to happen. But we use Cre recombinase enzyme, and this enzyme allows site-specific recombinations at where sites where there is LOXP. If you add LOXP into the DNA sequence, this Cre recombinase will identify this LOXP. Okay, and only there the recombination process will happen. Now suppose you have a transgenic mice, okay, where you have added LOXP sites like this, and in between this is your gene of interest which you want to knock out. This, I'm representing L, okay, I'm writing L with this LOXP, and there is another mice, okay, transgenic mice again. This is another transgenic mice which is having the gene for Cre recombinase. This is the promoter. Now, when these two mices they are allowed to mate, okay, then in the in this in the embryo, in the resulting embryo, the Cre will be expressed. Okay. Now, this gene where you have added the LOXP, this gene you have selected in such a way that it only expresses, say, in mice's heart. Okay, and this gene is not expressing in any other organ of the mice, and you have selected this gene in such a way while adding the LOXP sites. If you're following, then it's fine. Okay, till now I think it should be clear. Now, when these two mices, these two transgenic mices, when they are mated, okay, in the embryo, this Cre recombinase, it is expressed because of the presence of this promoter, and this Cre recombinase, if it is expressed in any other type of cell, okay, that is not heart cell, then it is not doing any function, like it is normal, it's just another enzyme, means it will not cause any DNA mutation at all, it will not cause any deletion or addition. But if this Cre recombinase, with the presence of this promoter, now this promoter you can choose to be again tissue specific promoter, okay, that there is a transcription factor, transcription factor which is only expressed in, say, heart tissue, and you have chosen the promoter in such a way that that transcription factor can only bind to here. So then you're also controlling where this Cre recombinase is supposed to express. Okay, now you, since you want to work with heart cells only, so you have chosen a promoter which has a transcription factor which is expressed only in the heart cell. So in the heart cell, the Cre recombinase is expressed. Okay, due to the expression, it will act on the log sites. Okay, and the recombination always happens from 5' to 3' end. Maybe delete this. So here, what will happen? This is your log site. It will turn around like this, and recombination will happen. So this is your gene of interest. Okay, so because of this recombination, this gets ligated with this. So ultimately, you will get one gene like this, and this segment gets out, deleted. This segment gets deleted. Okay, so you're causing deletion of a DNA segment only in a particular tissue type by using the Cre and LOXP sites, Cre recombinase and LOXP sites. You are doing your engineering, your knockouts, which are conditional. So the in animals, so these kind of animals will be called conditional knockouts. I think it will be clear. Okay. If anyone has any doubt, please let me know. Maybe I have, told something wrong which you may not agree with, or if you have missed any point. Is there anyone like that? or is it clear to everyone what is happening, or like how mice models are created? So,
So the
hmm,
sweep process of the conditional knockout, can it be done in a single generation by using a single mouse, or it should be crossed with the two mouse, then it can show its effect?
so I think you're asking is whether this uh, conditional knockout thing, if once I have created a mice where there is conditional knockout, whether it will be
No,
passed on to next generation. Is your question?
let me explain it. So
Okay.
suppose conditional knockout mouse can be done using the LOXP model, right? So
Mm-hmm.
in one mice we have added LOXP, right?
Mm-hmm.
And another mice we have added the Cre recombinase
Mm-hmm.
model, right? So
Mm-hmm.
we are gonna cross the two mice, then we can see the effect in the next generation that the particular gene is going to be deleted, or
Yes.
it be done in a single generation. Did you get it? What I'm trying to say? In a single generation, in using a single mice, can it be done, or we have to wait for one generation to see its effect?
Okay. So you are saying why not add both clocks and Cree in the one mice and see, right?
Yeah, yeah.
Okay. So for doing that, let me think about it. See, you have to add two transgenes, right, in a particular embryo in order to see uh, that how that embryo is developing. Correct? You have to
Yeah.
add two genes. Now, what is the possibility that all the all the stem cells, okay, get successfully transducted, right? It is repeat experiments. It doesn't happen in the one go. You have to do again and again. So when it is so difficult in even transfect and get the phenotype for one gene, you are saying that add two genes at a time. So I think theoretically it should be possible, okay, but success rate is much more higher uh, if you work with one gene. You getting my point? If you wait for the next generation, the success of getting conditional knockout increases, and these experiments doesn't you know happen in few months. It can take years. So you have to choose the option that is going to give the positive result at the end of say 3 years, 4 years. Okay. So that's why people prefer to choose two parents and transgenically means add separate genes in two, because people have seen it is working, right? Theoretically I think it should be possible, but I'll still look into it, okay, if people have done it and how much time it takes. It's a good question, but right now concretely to answer that I need to read more on it. I'll tell you more on this in the next class. Okay.
Yeah. Mm.
Okay. Anyone else have any question? Okay. If not, let's quickly do four problems. Okay, we are running short in time. First is shown below is a schematic of a genomic segment and a gene targeting construct being, I think spelling is wrong here, ready for the generation of knockout mouse. Okay, identify the segment that is likely to be integrated in the genome if the construct integrates into the genome via homologous recombination. So here we are trying to create a knockout mouse. Here I think global knockout they're saying. As you can see, this is the sequence. There is no cree or anything like that. This is global knockout. So you have to identify the segment, that is segment here. Okay, which segment? 1, 2, 3, 4. Which segment? This is a target construct. So which segment in the target construct is going to get integrated in the genome? So what do you guys think is the correct answer? Archie, would you like to try?
Neomycin resistant.
Correct, you're correct. So when we do
So
knockout,
without
yeah,
without the toxin in gene, the gene which codes for toxin, everything can be integrated.
correct, correct. So since these are, allelic, right? So the sequence are similar. So they will also, some part of flank L will also get incorporated when we are doing, recombination here. So neo R, exactly the neo R, actually that's a new segment that will be integrated and exon will be deleted. Okay, so answer C. Answer is the option C. Now, with next question, which of the following statements are incorrect pertaining to the transgenic model of a human disorder? We have to identify the incorrect statement. They contain exogenous DNA. They can model chromosomal disorders. They represent a gain of function mutation. They represent a loss of function mutation. The correct
D.
answer would be D. You're correct. So they don't represent loss of function mutation. Okay. Next, knockout mice models are best suited to study which of the following? One, haploinsufficiency, then loss of function mutation, recessive disorders, all of the above. What do you think is the correct answer? So haploinsufficiency, if you recall, it often happens in heterozygous condition where the even though the mutant allele is not functional, but whatever protein the normal or the wild type allele is creating, it is not sufficient for the proper functioning of the cell. So even if the normal allele is present, okay, the cell is not showing the normal phenotype. Sometimes it needs sometimes protein should be expressed from both the um, normal should be expressed from both the normal variant of the allele. Okay, the double protein is needed for functioning of a particular biochemical reaction. If that is not happening, only half of the protein is present in the cell, then a particular phenotype can result. That is haploinsufficiency. So what do you guys think is the correct answer here? So the correct answer is option D. Okay, all of these. We can study all of these by using knockout mice. So we can create a haploinsufficiency kind of a thing, right? Say we can create the mutant allele by deleting a particular segment. Okay, and so in the mice there will be the mutant allele which is not having a particular segment of the gene, so it is not functioning properly, and it will also have a normal allele. So we are trying to create that haploinsufficiency kind of a condition in that mice. Then obviously we can study loss of function mutation. Okay, um, and we can study recessive disorders. We can create deletion in both the copies of the genes and thus create a recessive genotype. Okay, so in all of these conditions, we have to just delete a particular DNA segment, and knockout allows us to do that. Okay, maybe for recessive disorders, we have to, do for several generations, then only we'll get the required phenotype because we are deleting segments from both the copies of the gene. So it will take a bit more time, but it is possible. Then last question. Have you guys understood this question? Why it is D?
Yeah.
Okay.
Can you turn the slide two slides back, because I have to see the question once. That
Okay.
just two slides back. Question number 13. Yeah.
This one, no? Okay.
Yeah.
Okay.
Yeah. Okay. Thank you.
Okay. This is the last question for today's session. So, which one of the following statement is incorrect with regard to a conditional knockout strategy used in mice models? Okay, you have to identify the incorrect statement. They can be used to model monogenic disorders. They can be used to model loss of function mutations. They can be used to study gene functions, and they can be used to model gain of function mutation. Which of these are incorrect? Which of these, one of these is incorrect? You have to identify which is the incorrect statement. Can you guys
Yeah,
answer? D, yeah, okay. Yes. Knockout, any like even conditional knockout cannot help us to study gain of function mutation. For that, we have to create a transgenic mice model. Okay, whereas we can study the monogenic disorder, which is nothing, where a disorder is due to defect in one gene. Okay, and also there are polygenic disorders where defects in multiple gene result to a particular syndrome or a particular disease. And yes, we can study loss of function, and yeah, we can study gene functions by understanding what would happen to the mice if this gene was not present, and then we can interpret, okay, now if this gene was not present, this was not happening, so maybe this gene is responsible for this function to happen in the cell. So these kind of studies we can do and identify the gene functions. So that's it for today's class. I hope you have understood the content which I have, explained it to you. Thank you guys for joining, and I'll see you in the next week with the week 4 content. Okay. If you have guys registered for the exam, then all the best. I'm ending today's meeting. Thank you.
Thanks.